Hi! These slides cover some of the basics of zoonoses and vector-borne disease, as well as some of the environmental factors that influence disease transmission. Check the text for more details on specific zoonotic and vector-borne diseases. According to the World Health Organization, zoonoses are diseases and infections that are naturally transmitted between vertebrate animals and humans. A zoonoses can be any type of infectious agent, like a virus or a parasite, but it must be transmitted between humans and vertebrate animals. Usually the term zoonosis refers to transmission in the direction from vertebrate animals to humans, but the infections can also be passed from humans to vertebrate animals. This is called reverse zoonosis. About 60% of all human pathogens are zoonoses. They account for 75% of emerging pathogens in the last few decades. Examples of zoonoses include Salmonella, Toxoplasma, SARS, and avian influenza. Zoonoses can be transmitted in multiple ways. Fungal infections are an example of infection caused by direct skin contact with an animal. An example of a disease transmitted by inhalation of a pathogen from environmental sources is hantavirus. This virus is present in rodent droppings. Humans are exposed when contaminated dust is disturbed, such as when cleaning out a barn. Commonly, zoonoses are acquired through ingestion of contaminated water or food. Most foodborne illnesses will fall into this category. Contact with animals, such as at a petting zoo, can result in infection because of hand-to-mouth activities among children. Think of thumb sucking. Rabies is a well-known example of a zoonosis transmitted through the bite of an animal. Finally, zoonotic disease can also be passed via an arthropod vector. Plague is one such example in which Yersinia pestis, the bacteria, is spread from rodents to humans via fleas. A disease that is transmitted by a vector is called a vector-borne disease. A vector transmits pathogens from one host to another, which could be the same species, like from one human to another, or different species, like from mouse to human. When we talk about vector-borne diseases, usually we're talking about arthropod vectors like mosquitoes, ticks, flies, and fleas that transmit pathogens through a bite or a scratch. Technically, vectors can also be rodents if they carry a pathogen from a reservoir to a host, or even inanimate objects that can carry disease, also called formites. Vector-borne diseases can either be zoonotic or not. For example, Lyme disease is a zoonotic vector-borne disease because the disease is transmitted from vertebrate animals, like deer mice, to humans via an arthropod, the tick. Zika is a vector-borne disease that is not zoonotic, as it does not appear to have an animal reservoir among vertebrate hosts. Interestingly, there are several species of malaria, one of which is considered zoonotic, while others are not. An emerging zoonosis can be one that is newly recognized or evolved, or one that has occurred previously but shows an increase in incidence or expansion in geographical, host, or vector range. This can also apply to vector-borne diseases. For example, dengue, Zika, and West Nile virus are all vector-borne diseases that have expanded their range over the last 2 decades. There are multiple factors that drive zoonotic and vector-borne disease. The rest of this presentation features several of these factors which are the focus of the elective public health class 1 Health Zoonoses. Poor health, sanitation, and housing infrastructure can leave people more susceptible to zoonotic and vector-borne diseases and the health consequences of these infections. Similarly, poverty increases risk for zoonotic disease, but the reverse can also be true. Disease in humans and/or livestock can significantly affect livelihoods. Many zoonotic diseases are considered neglected tropical diseases. The term neglected highlights that disease affects mainly poor and marginalized populations in low-resource settings. War, conflict, and displacement often result in a crumbling of infrastructure and can increase the risk of infectious disease, including zoonoses. High-risk areas for disease emergence and reemergence are those in which population growth is high, development is disruptive to ecological systems, and where human and wildlife populations overlap. All 3 of these characteristics may be present in rapidly growing urban centers, primarily in low-income countries already burdened by zoonotic disease. This is a CDC photo from Kibera, a slum in Nairobi, Kenya. Humans have been living with dogs for about 14,000 years, so it's not a surprise that pathogens have evolved to affect both dogs and humans. While canine rabies has been eliminated in the United States thanks to vaccination, dogs account for the vast majority of rabies cases worldwide. Cats are relative newcomers on the domestication scene. They were only domesticated about 4,000 years ago. They are less frequently the host for zoonotic diseases, but they are the necessary host for Toxoplasma gondii, the parasite that causes toxoplasmosis, which can be dangerous for developing fetus and those with compromised immune systems. This pathogen is the reason why pregnant women are advised to avoid scooping the cat box. More recently, keeping chickens and reptiles as pets has resulted in salmonella infection, particularly among young children. The effect of climate change on zoonoses and vector-borne diseases is difficult to predict because of the multiple factors and the interactions among them. These photos, clockwise from top left, represent the ecosystems, hosts, vectors, and pathogens potentially involved. Furthermore, climate change is not just one thing. This is why the term climate change is preferred over global warming. We will see changes in temperature, precipitation, humidity, carbon dioxide level, sea level rise, and extreme events, which will vary by location. Anthropogenic change, such as land use, pollution, and the introduction of new species, further complicate the equation. The photos are a rainbow over the jungle foliage of the Guam Mountains, a deer mouse, which is the principal reservoir of Syn-Nombre virus, which causes hantavirus in humans, a black-legged tick, which transmits Lyme disease, and an electron micrograph of yellow-colored bacteria on a flea. These bacteria cause bubonic plague. Other factors that influence zoonoses and vector-borne disease include global travel and trade, antimicrobial use and food production, and changes in agricultural practices and in food processing techniques. Contact between humans and animals or vectors can also result from the encroachment of agriculture into habitat. Agricultural land use practices can also reduce biodiversity, which can affect disease transmission cycles in unpredictable ways. There are many environmental variables, mostly highly influenced by humans, that can change the dynamics of zoonoses and vector-borne disease. These complex interactions require a One Health approach in which the health of humans, animals, and the environment are considered together.